Okay. Okay. So hello, everybody. So welcome to the Badger Dairy Insight. I'm Alison Fao. I'm the bilingual regional dairy educator with UW-Madison Extension covering Dane, Jefferson, Rock, and Walworth counties. This is our monthly webinar series providing the latest information research to help producers, workers, and managers. Today's topic is going to be how nutrition helps us in taming inflammation and improving cow health. And with that, we're going to have two speakers, very special speakers. It's going to be Dr. Eduardo Rico from the University of Pennsylvania and Dr. Daniel Rico from CRSAD in Quebec, Canada. Could you please, Daniel, tell us what is CRSAD, please?
Hello, Alison. Yes, this is the French wording for Research Center in Animal Science at Deschambault, which is the little town where we are. Very close to Quebec City in the province of Quebec, Canada.
Okay, thank you. So I'm going to introduce themselves. So Eduardo Rico is an assistant professor at the Department of the Clinical Studies at the University of Pennsylvania School of Veterinary Medicine. Eduardo studied animal science at the National University of Colombia. So, woohoo! And after working for several years as a nutritionist in the feed industry in Colombia, he moved to the US to complete his master and doctoral studies at the Cornell University. His research focused on how nutrition influences dairy cow metabolism, physiology, milk production, and health, particularly regarding fatty acids, energy use, immune function, and peripulture ketosis. Daniel Rico, his brother, and now here from Bogota, Colombia. Also, he holds master and doctoral degrees in animal science from Penn State, specializing in nutrition, metabolism, and milk quality. After postdoctoral research at the Laval University in Canada, he has been a researcher in animal nutrition and physiology at CRSAD since 2017, investigating nutrition impact on animal health, productivity, and the environment through a One Health concept. So thank you so much for being here. Please, for the audience, use the Q&A button for questions. This webinar is going to be recorded and will be posted at the dairy website at the UW-Madison Extension. So you can find all of these videos right there. So thank you so much again for being here. We appreciate that you accept this invitation. I'm very happy to have you two here because we are from Colombia and I'm very, very excited. So the floor is yours and we can start now. Thank you so much.
All right. Well, thank you, Alison, for having us. It's exciting for us also because it's the first time that we do this sort of thing together, as we talked about before. And we go way back, Daniel and I.
You could say that, yes.
You could say that. So yeah, for those who haven't realized yet, we are twin brothers and we are both interested in nutrition, in animal nutrition in particular, dairy cow nutrition in particular. So there is a lot of overlapping with what, and you can imagine with similar brains, the things that we're interested in doing. So as you will see today, there's a little bit of stuff that we have in common that we actually are happy to discuss here, and that relates to how nutrition can do something about health and welfare. And the common theme that unites us today is inflammation, right? Without any of us being immunologists, just trying to dip our toes from the nutrition angle. But probably that's kind of like the disclaimer that I will have to start. So yeah, let's get it rolling, Daniel.
And I wanted to say, yeah, maybe we can go into questions at any time. As you get the questions in the chat, in the live chat, Alison, we can go into that at any time.
Yes, of course, I will do it.
All right, so maybe Eduardo, this would be a good spot to start explaining why we're talking about inflammation today. So it seems like it's a topic that many people are talking about for a long time. So yeah, you have a few examples here. Maybe you tell us why this is important to you.
Pretty much in the popular culture. We were just discussing this before we started recording. That everywhere you look at, and this is an example from the past couple of months, just growing grocery shopping. We talk about how inflammation is something we need to control, right? Or in this case, that's probably a word that we should evaluate more closely. You don't want to stop inflammation. We're gonna probably go into that, but the point is that there is something about inflammation that we want to avoid. right? And so what do you do with diet in particular? Do you eat more of this, more of that, this particular supplement? So there is a bunch of parties interested in this, the ways in which exercise also could help. But in the human world, this is a how do you call it, a fledgling, burgeoning field that is very, very pretty much in anyone's interest. And of course, that's also what you could think about animals. For us, there being an interest in this area. So pretty much in the popular culture, yeah.
Yeah, this is something I see also here in Canada. There's, well, also a bunch of podcast people on YouTube, everywhere on X, talking about inflammation. And there is a lot of claims. So maybe there's some truth to the importance of this, of course, and misunderstanding a little bit if this is a good process or is a bad process. Because to me, kind of that was like something that a few years ago we started working on. And maybe the first confusion for me was, okay, this is good. Why do we have to control it? So I think you have a good analogy for the tree forest, the forest fires.
Yeah,
Maybe let's
let's
go
go
into
to that. But let's preface that with saying that inflammation is a necessary protective response of the body to things like injury or infection, right? So there is an insult, and to that insult, we have an army of cells that are ready to respond. So here we actually, in this slide, we could see two groups of cells. On the left side, red is the ones that are in charge of leading the charge, just to be repetitive. Some cells that are taking care of is the first responders, you could think. And then you have some others that are there to put out the fire that was started, right? So you have an inflammatory response that you can think of as a fire. And I think about this when I think about the Barry Bradford's review 10 years ago. He puts the fire at the center of this, right? And it's important to keep the health of the animal. That's the long story short, right? So these fires, the analogy that I like to place is what people in California, places like California, think a lot. You do a controlled fire and you contain it so that you can improve or maintain the health of the forest. So in the same way, you could think of letting inflammation or the fire take place, but in a controlled manner, in time, controlled in time, and controlled in intensity. Those two things need to be controlled, right? When it goes out of those bounds is when we start having issues. So that's the shorthand for the analogy, right?
So the analogy, if we take it back to the animal, then it is, let's say, the naturally dense forest is having all of these microorganisms that shouldn't be there, for example, during an infection. The body is going to deal with it, and the response can be too much or too long. So chronic inflammation is an issue. We talk about that a lot on the human side. So for the cow, I guess that's the thing. We need to resolve that inflammation. And there are
Yes.
ways that we can do it. That's what we're going to talk about today, to tame it and then resolve it, which are two things that are not exactly the same.
Yes, and I would say that as a preface to that, that also one of the opportunities we have with nutrition, and we will get that into that part when we talk about the omega-3 fatty acids, is that you could do something with nutrition to empower the cow to mount a better resolution of that inflammation, right? To put out that fire, which is something that not necessarily pharmaceuticals can do. Pharmaceuticals can act on a particular area which is blocking the inflammatory response or reducing it in intensity, but not resolving it, which are two different parts of it. Okay, so yeah, we'll get into that as we go.
Yeah, maybe now what we could mention also is there is this danger of having uncontrolled inflammation. But what is it exactly for the cow? What is the problem that we have with that? And you have this list here of things that are happening: tissue damage, appetite reduction. So what are the
So
highlights of this?
So there's the things that we see and the things that we don't see. What we see in the end is that animals will produce less milk, will have impaired fertility, right? Low reproductive function, which are the outcomes. The cow tells us that there is something wrong, but what we don't see is what happens before you get there, which could be weeks or months. And that's damage to the tissue because of this inflammation that was not controlled. And in the case of the mammary gland, you can think of fibrosis of the mammary gland and tissue that doesn't go back to being, the milk secreting tissue that should be. And you have a number of things that are happening to the cells themselves that are damaging the cells for the longer term, and are also going to have an implication on cow longevity or productive life, as we say, because the cows are going to ultimately be called out of the herd early because of these things that we don't see. And I will make emphasis on the things that we don't see because that's that goes to the topic of markers. How do you know what's happening inside of that cow? And other than just looking at the obvious signs of milk yield or appetite, right? You don't really know much because it's hard to measure. So there you
This
go.
makes me think of something that we will discuss later, but we have been working on heat stress for a few years now. And we got to it on the side of cow productivity, milk quality, etc. But then discovered, of course, that there is all of this inflammation happening. And I think this is a good summary of what's happening during heat stress as well. So there may be some cell function at the mammary level, the gut level, that there is a loss in that function. So things don't start going really bad. Then the animals reduce their appetite, they eat less, but eating less doesn't really explain everything that is happening during heat stress because there's more to it. There's the inflammatory response, of course, all of that energy, and then that energy going into the immune cells and not to milk synthesis. So that's something to me that was kind of an initial interesting discovery is that, wow, heat stress is really a variation on a different flavor of inflammation, but it's kind of the same thing. The same things are happening during heat stress.
So
So
you would
getting to that
do,
point, yes,
if you could do something about that response, so that is not so extreme, or that the cow is, I would say the word empowered again, better prepared to deal with that, then you could do something about how that energy allocation to the immune system, which is kind of like a loss, you could say, for in terms of milk production, could be avoided. But bringing us back to the importance of trying to assess inflammation or prevent inflammation or do something about inflammation, because ultimately it also means not just that the cow gets sick, but that she's going to make less because there is less nutrients to go around.
exactly. And then just rounding this idea that many things can cause inflammation. We have the example of heat stress. The idea there is that you're going to have leaky gut because the barrier, the intestinal barrier is compromised. So you have these bacterial origin compounds such as lipopolysaccharide that we have here in the picture, LPS for short. That's going to trigger the response. There's also mastitis, metritis. So any infection that the animal has to deal with is going to result in that type of fire. And then the response of the animal, this is something you could probably go into deeper, is we have this primary response with cytokines, and then some of the cytokines like interleukin-1, 6 are going to trigger the secondary response, which are the acute phase proteins. So that's another
Yeah,
thing about the markers. These are markers of inflammation. What is the thing with the markers?
this is the thing that, again, that we don't usually see, right? And these things happen very quickly after there is an insult. So, for example, the cytokines are produced in a matter of hours after there is kind of like a breach, say that the LPS enter into the blood, or that there is an actual infection, right? So these things happen quickly in hours, and if you could time it and measure it there, you will know what's happening, which we don't usually do, of course, unless you're doing research. But then there's the longer response that could take days to appear, and that's secondary in the sense that these signals get into the liver, and the liver is actually going to produce these proteins that we call acute phase proteins. And probably you hear more and more of those because they are actually very good markers for some things that are happening chronically in the animal, so over the course of days. And it could have been in many cases over the course of weeks that the cow is dealing with an issue. And they could be actually helpful. I'm sure that you have and myself have measured some of these in our research. It's hard to find good ways to measure them. The tests don't always work. We all suffer with
Yes.
this ailment, but things like haptoglobin, C-reactive protein, LPS binding protein, they could be good ways to use this as proxies or indicators of something that is going wrong. And I understand you have used this actually when you're talking about heat stress, right?
Yeah, definitely. C-reactive protein, LPS, binding protein, and then the cytokines. All of these are elevated after a few days on heat stress. Well, at least it could be earlier, right? But our response is normally day 7, day 14. We have really high levels of this and fever. So on top of the heat stress, there is the fever response that is related to these cytokines and acute phase proteins. That's another thing that we can tackle with nutrition, and we're going to talk about that. And now
So
maybe
let me go back to this before I forget, because this relates to the biomarkers and what we're showing here with ketosis. So one point that we will probably drill on today is that there is a lot of things happening in the cow that are muddled, right? Or muddled, I should say. They're happening together and you don't really know who's the culprit. And to some extent, that tends to happen a lot with things like ketosis, we can argue, which sounds a bit blasphemous to say. to some years still. But these things that you see in the list are markers, basically indicators that something is happening. And I will emphasize again, invisible to many of us, right? Because they could be happening now, but you see the consequences 3 weeks, 4 weeks from now. In the case of ketosis, some of these markers are actually interesting because this data came from Michigan State, and you can add to this if you want, Daniel, but the majority of cows that are diagnosed with clinical ketosis appear to display or to have this endotoxemia. So these substances of bacterial origin, presumably, I understand, come from the gut. They just filter into the circulation where they shouldn't be. And they are surely causing some issues, right? So it brings us to the question, are these cows displaying all of these issues that we call ketosis as a result of the ketones themselves, or actually because there is infiltration of things that shouldn't be there, and they're of course causing damage, reducing energy supply for the cow for milk, etc., etc. So there's something to be said about things that are happening simultaneously, and we don't really are able to measure the right thing on time, right? I will also add here that those ketones are something that we measure when the cow is actually very sick, and it's what we call the negative energy balance state. So what if we could measure these markers earlier, which some people are trying to do?
So with that, I have a question. So you say that some of the inflammation tests are not working or they are not good enough. So how you can improve them, or what is missing that those inflammation tests are not working?
All right, I don't know, Daniel, go ahead, tackle this one.
No, I guess it's a technical issue that sometimes we have to be careful when we use this, for example, for research. We have to make sure that the antibodies that are made for detection of this are actually well developed for the bovine, and there's an issue, a technical issue there. But then there's the other question of using this at the farm level. If we compare them to ketones, things that we can measure really easily, like ketones, we don't have that yet for the dairy cow on the inflammation side. So I think, Eduardo, you're aware of some work that is being done on that?
Yeah, so the latest I've seen, and this came recently in the Journal of Dairy Science, there is some work at Cornell at the vet school, which is, I understand, done by Sabine Mann, Dr. Mann, in collaboration with some other people. And they're trying to develop these immune tests so that we could use, hopefully, at the farm someday. And of course, she will be the best person placed to talk about this and explain this. But as far as I understand from our short conversation recently, it is a big challenge. So it's not only challenging for us to measure these things in the lab, but it's challenging to actually make them work in an easy cow side test at the farm. The advantage that I see in the future, if we get to overcome that hurdle, is that we will be able to do better diagnosing animals that don't have, these issues late but early, right? So you detect them pretty much pre-onset, before things go south, when you could do something. So that's kind of like the challenge that we have right now when it comes to measuring these things. All right. So hopefully that answers the question. So this is the point that we wanted to make about biomarkers, right? Indicators and ketosis, because we're going to talk about a little bit about ketosis after this.
Maybe now it's a good time to go into why that increased immune response is kind of a drain in the fuel economy. Let's call it like that. So we actually have these slides that we discussed the other day. So if we just take the example of glucose, that glucose that is being used by the immune response to face this challenge is going to go there. There's good data from Lance Baumgart that we also have in the slides that the immune cells are going to use a lot of that glucose when there's a challenge, an LPS challenge, for instance. Is that's something that is really important, because it's not going to go to the mammary gland to make lactose and to make milk. It's not going to go to muscle, to the ovaries, to other places, because there's a priority there in facing that challenge. So it's something also that is going to affect metabolism. We know that the immune cells can talk to the pancreas and tell the pancreas, please release more insulin. When there's more insulin, that's going to allow the immune cells to use that glucose, and it's going to do the opposite for the mammary gland, because the mammary gland doesn't really respond in the uptake of glucose to insulin.
So this is what people talk about when they talk about differences in partitioning of nutrients, right?
Correct.
Where is the energy or the nutrients, I should say more generally, going? Are you gonna close the tap to the mammary gland so that they can go to the immune system, to the defense army, to feed the army, I guess? And so if that's happening, you're gonna reduce the flow to the mammary gland. And of course, you're gonna have less milk. That's kind of like the short explanation, I guess.
Yeah, that's a good way to say it. Like the water tap. It's going there, but it's not going there, or it's closed. So we're gonna see effects in metabolism, in productivity, etc. So I guess that's the only to say there's a lot of interest in the inflammation story because of this angle as well. It's not just the damage that the cytokines or chronic inflammation could do, but the use of fuels and the productivity of the cows and how we look at their well-being and all of these things.
Yeah.
And
So
you have this data here that maybe we can go into. If we're talking about that glucose that is not going to make milk. So where is it going? So we have some data here to show that.
yeah, this is from a couple of groups, Lance Baumgartz from Iowa State, and then Professor Trevisi at the Catholic University in Italy. I've been working on this area for a while now, I would say. And what Baumgart's data showed us is pretty interesting in that when you activate the immune system, so imagine there is a challenge, like an infection. In a 12-hour period, they showed that there could be around 2 pounds of glucose being consumed in that period of time, which in the course of a full day is around 65 pounds of milk that are lost, right? Because that energy for 65 pounds of milk could not go to the mammary gland to actually synthesize the milk. The other example is the peak milk yield in lactation, right? So when cows reach the peak, that's around 10% lower in cows with increased inflammation. This is the Italian data. So inflammation, even if you don't see any other obvious signs, it's silently reducing the performance because those nutrients are being, diverted to feed the army again. So that's kind of like a very easy example of why this is relevant. How much milk is being lost, how much money for farmers is being lost ultimately.
And on top of that, there is the reproduction issue. And I think you also have this data set that illustrates really what's happening with fertility.
So yeah, defining inflammation is kind of difficult if you use just one thing. But these markers, I think that you could make the case there are very good indicators that there is inflammation happening, especially chronic or over the long period of time. In this case, HP stands for haptoglobin, which is one of those things made in the liver in response to a challenge or an insult. And when these cows in the orange have an elevation in that haptoglobin above this threshold, they show two things: reduced fertility, which obviously you can see from that pregnancy fertility curve over there, cows not pregnant. So there is more cows not pregnant when there is high haptoglobin. And there is also the consequence on production, which is around 1,000 pounds less over the course of a 305-day lactation. So significant, pretty major, and not necessarily something you will obviously see unless you are tracking these type of markers.
Now that we have discussed the issues with inflammation and the immune response being one of the places where the energy or the glucose can go, maybe now we could talk about this confusion that we may have around what causes disease in cows. And that's something that a lot of people are talking about, have been talking about for a few years now. And again, you have some work that I really like this review that you guys prepared. And I guess this is a good point to say, to illustrate that ketones are part of the equation, of course. This is something we should be paying attention to. So that, let's say, blasphemous statement that ketones are not important is not necessarily true. And I guess there's just an integration to be made with the immune activation, inflammation, insulin resistance, all of this. So what was the highlight of your review here? And
I think that
after that, let's discuss observational and control research, because that's something that you also talk about in your review. I think something that we don't often talk about, and we should really talk about that to understand why this can be a complex issue.
Yeah, no, not all types of science are equal, and what we interpret, it could be in the human world and in animal science world. The type of research that we use to back our statements is important because it tells you something about how much confidence you can put in those statements. So you made a good point with the ketones. I'll try to make it short. Of course, you're invited to read the review that we made on this, what we call a changing paradigm. So the idea that we have about ketosis is being modified, I will say, is not being put down necessarily or put to rest. It's just that we posit here that there is more to it than just the ketones, and ketones are actually something very easy to measure. So when you When you can measure ketones, everything is gonna, I mean, it doesn't surprise me that you're gonna call anything that is related to problems, when you measure them, you're gonna call ketosis, or it becomes a bad word, right? So in many people's minds, still a bad word. I will briefly say, in the human world, this used to be a bad word uh, decades ago, and it transitioned briefly to the really cool thing that it reduces chronic inflammation, that helps you lose weight, that became like this amazing thing, ketogenic diets. And yet in the cow world, we're still thinking on this very unique tunnel vision way sometimes, that it has to be bad. So that preface to say, there is much more, as you can see in this diagram, happening in a cow that is making milk than just the ketones, right? There is a number of things that could be causing issues, could be causing insulin resistance. We could argue about that, if that's an issue or not. Oxidative stress, cellular dysfunction, things that ultimately make the cows be less fertile or make less milk. Inflammation, you could see it also there, is one of those things that we don't measure usually and tend to happen at the same time. So imagine that you have inflammation with all the negative effects that it has. And at the same time, you have ketones, but you can measure only ketones. So you say, okay, ketones is an energy molecule. Let's fix that. Let's fix the energy issue. Is it enough to give propylene glycol, or to give dextrose drench or something, 500 grams? Is that enough in the sea of, in the ocean of the drain that you have on glucose itself, as we showed before, that equals 65 pounds of milk per day? It's very hard to make a dent on that when you are focused on fixing the ketone issue. So that's kind of like the long story short, of why we should care about more other factors that we cannot usually measure other than just the ketones.
Okay, so you guys are talking a lot about like cows, like the peripartal cows.
Mm-hmm.
What do you think about the calf? They also have some inflammations and some issues and some disease. So what about them?
Wow, this is a a concept that would apply to any type of animal in general. When you have an insult, this is my crack at the question, Eduardo. You can complement later, but if there happens to be any inflammatory challenge, immune challenge, the animals, especially around the weaning period, are going to experience in early life in general, depending on the conditions where they are, etc. The environment can also put a lot of stress on the animals. So they are going to experience inflammation. And one of the things we're going to see later that is true for the dairy cows, but also for the calves, is that we're not really sure about the requirements for these periods of challenge. And I will have to be more familiar with the calf literature, but I guess the principle applies to any period of challenge at any moment in an animal's life. So if there's going to be immune activation, is that going to be controlled? Is that going to be optimally like disposed of when it's really done its job? So the resolution part, all of this that we can talk about with omega-3s, is that something that would be of importance to the calf? Yes, definitely yes. Now, the work that has been done, I need to be more familiar with. So I guess we'll
That's
get
fine.
back to
That's
that
fine.
at some
That's a
point.
good crack at it. I mean, obviously, we don't really work so much specifically on calves, but I will say something. We do work on early life stress in general, and we have some work also together doing some work on models that reduce inflammation during critical life periods. So we started with the piglet model, and then we want to graduate into calves. But the idea is overall the same, that when you are presenting the animal with a very challenging situation, which is, for example, going from mother's milk into the concentrate feed and forages at weaning, there is this apparently unavoidable slump, fall in growth rates, and the animals are not doing great. And we talk about weaning stress. Now we could discuss to what extent that could actually be defined as weaning stress, but there should be things like inflammation happening. So that would be the common thread that I see into this. Those types of stresses could, to some extent, apparently based on what we have seen in other species, alleviated with nutritional strategies. There may be others that are not so easy to tackle. So if we're talking about a viral infection in a calf, or we're talking about a bacterial infection, that's a whole other kind of worms, right? That is not necessarily so easy to tackle. But I would say that there is something to say about preparing the animal or arming the animal with everything you can to defend itself against the regular insults, given that there is good management, right? Because it doesn't work if there's no good management. So that would be probably the way that we could kind of answer the question. All right. So this whole thing was just to illustrate that there is a number of things happening, and that we're going to probably migrate to the one thing that we could measure. So one of the solutions is measure more things, right? So we will start with saying inflammation, which is the case we're trying to argue today, is important. It's an important part of this equation, and we should be able to do more about measuring and detecting it, hopefully early enough that we could prevent downstream issues, right?
Let me
So
just cut you there for a second. In the interest of time, maybe we should move before we go into the nutrition part, because that's what we do in nutrition. Let's just round up this idea that there is a type of research that can lead us to have some answers, and that we have to be mindful when we think of the types of research. And this is something that is part of your review. So I don't want to leave that or forget about that when we talk about this. And for some reason, that slide is not showing, but the point that I wanted to make here
no,
is that,
here you have the shark. We wanted to talk about sharks today.
yes,
That's good.
well, when we compare observational research versus controlled studies, controlled clinical trials, we have the issue that we cannot control for everything in the observational data. So we just look at the world, how it is, and we try to draw conclusions from that. Let's say from an observational study that can give us a lot of clues about things that are happening. We don't really get all the time cause effect. Sometimes we could, sometimes we don't know really, right? So I
Yeah, we
guess
get
the example,
relationships.
right? The example here with the shark is kind of a fun example in my opinion, because we get when we get to the summer months, there's going to be an increase in ice cream sales, of course, and also in shark attacks. But we already know intuitively, looking at this data set, that even though the correlation is super strong, is not really true. Like you're not really more at risk if you eat ice cream, if you think about shark attacks. So it is intuitive, we know, but it could also apply to anything else that we just measure. For example, ketones going up when the animal is sick. And it could prevent us from understanding what the real cause is. So maybe, as you said, we're measuring ketones. That must be the culprit because the animal is sick. But those ketones could be also elevated in an animal that is not sick at all and is producing a lot for different reasons. So how good is a marker like that when it's present in healthy and not healthy animals, right?
Yeah, we're gonna get to that in terms of research, but before that, I will say that this is one of the reasons for which I don't eat ice cream when I go to the beach in the summer. I just
We
want to
have
be
to be
less
careful.
tasty for the sharks.
Yeah, good point.
So that brings us to the observational research. So we want to try to close this with a bow tie here. Observational research is telling you A is related to B, and controlled research is telling you A causes B, unequivocally, right? So we try to, most of the time, as much as we can, do randomized controlled studies or RCTs or controlled research, where you change one little factor, only that factor, and you kind of like block the rest from influencing your outcomes. So you want to know, for example, if ketones are good or bad, or NIFA are good or bad, you change that thing in research. That's the beauty of research. You can control. But in observational research, you only can see, for example, these cows have ketosis, and at the same time, they have increased inflammation and increased oxidative stress. Chicken or the egg, which one was it? You cannot really know, right? So that could be good to guide us into do studies that we're trying to do. For example, I'll show you the example of ketosis now, where you actually say if that relationship holds. But that's an important thing to consider. You have this funnel, in this case, this soup, I guess, of factors all interacting together when an animal gets sick. And you measure one of them. And you do research on that one that you're observing, and then you make a disease name on that, right? So if we could measure NIFA, we would be talking about not ketosis or something more like NIFA-osis.
That's a good point.
Right? But it sounds facetious, but it's probably a fair point to say that it depends on what we can measure. So I will try to drill that point as much as I can. Inflammation could be happening at the same time that NIFA is high and ketones are high, and a cow is experiencing what we define fatty liver. Which one was it? Are all of them conspiring? Which one is more important? Hard to know. So what do? We go to do randomized research. So I'll show you now the example of the ketones just to illustrate that point.
So there's a few trials that you have run on this
Yes.
type of research.
So we're trying to change the ketone, particularly as a factor that we isolate and say, if we add the ketone to the body of the cow, is that gonna do something good or bad or neutral? And we have done a nutritional induction. So we create a ketogenic diet. That's one thing. Or we actually put the ketones directly into the vein, yeah, more like this, of the cow, which is what you see in the figure. So overall, this is just one example from all of that research. When we get to subclinical levels of ketosis, what we consider subclinical, that is supposed to have negative effects on the animals, we don't see any of that in terms of health acutely, at least over the course of anything between 24 hours to 5 days. We have done evaluations.
So that would be this blue line, correct?
That would be that blue line. This is just an example intravenously. And we didn't see any changes in the appetite of the cows during this time, or milk production, or anything of relevance, right? The actual markers that matter. Is the cow eating well and milking well? We didn't see that change, except when, and I put that in the title here, during immune activation. So it brings us again back to the topic of immune activation and immune system and inflammation. So when you actually challenge that cow with something that makes that immune system freak out, in this case, we call it the LPS challenge. We give them the carcass of a bacteria, and we make the immune system think that there is a pathogen, right? But there is really no alive pathogen there. It's just kind of the carcass. But that's enough to activate the immune system. And in that situation, when you actually need to have some inflammatory response, you kind of stop it with the ketones or reduce it with the ketones. So we may think that ketones may be conditionally important in terms of the peripartum in situations where you actually need to deal with some kind of insult, but not always. Most of the time, and this could explain why, a cow that is milking a lot could actually have very high ketones, right? So no signs of being not healthy, which is, again, paraphrasing Lance Baumgart, if the cow is milking well and eating well, is she sick, right? So this will probably support that statement.
One question about your model, because as you mentioned, there's different ways to do this control research. You could just give calcium butyrate and increase ketones like that, like we did. Here you have the intravenous infusions, which I think is a really good model. But isn't it true that you're missing the NIFA? Like a really sick cow is mobilizing a lot of adipose tissue and lactation. She has high BHB. The two are associated, of course. And it's not part of your model here. Is there a reason for that? Like, what do you think would happen if you add the NIFA?
Yeah, when you that's kind of like the issue. There is a great work done back in the 90s with Dr. Drakely from Illinois. The model that he used to induce ketosis actually increased NIFA and BHB at the same time. So that's good in terms of reflecting what a cow with ketosis is experiencing, because there's many things happening, as I said, but it's not necessarily the best way to pin down on the effects of ketones themselves. So we want to answer first the question, what do ketones alone do by themselves? And then we're adding, this is current research we're adding on top of that a pile of other factors. So ketosis plus immune challenge, ketosis plus NIFA, ketosis plus other things. So that's a question that we answer progressively, but we need to isolate factors. So that's probably, I don't know if that answers your question. But
Yeah.
we don't want to do everything at the same time, because then you cannot disentangle that web.
I guess one thing that comes to mind is that you can try different models and see where the data point, in which direction, and then conclude if there is a role of ketones or not. There's not one single experiment that is perfect for showing everything that is happening in a complex period of time, such as the
True. And that's the reason for which we titrate this in the mid lactation, but also in the early lactation. We try this with a nutritional model, and also try with an intravenous infusion. So different ways to try to see what answer we get as a convergence of data, not necessarily single isolated studies. It's tricky. This is not necessarily easy to do, but I think it's important at least to see if we should be moving in a different direction or a slightly correct course.
Yeah, of course, I think you're doing very cool research. So I guess I'm biased, but it is interesting data to see what is happening with the ketones by themselves.
That's called the twin brother bias.
Yes, it's a particular type of logical fallacy. So let's continue with the nutrition part. I guess we have something like maybe 10 minutes more.
Yeah.
Let's go into the nutrition part. So what is your...
Okay, big picture. We're going to switch gears here a little bit. Part of what we wanted to do was to set the stage to say inflammation is something we should care about. And of course, you hear not only from us, but from many people. But then what we as nutritionists care about, which is how can you help animals in terms of health and welfare, ultimately, to do better in terms of controlling that inflammation. So if we made a case that inflammation should be controlled, how do we control it? So here in this slide, you could see many ways in which you could try to do something about inflammation. A lot of that has to do with management. So probably I would just point out to forage and grage management is one of those things that you should do to actually provide the bare minimum from the nutritional side. And that's leaving aside things that include for example, stresses like heat stress that you should correct to some good extent with proper heat abatement, right? But let's focus on the nutrition. So other than forage and grain management, there could be other things. I put ketones there because they're actually anti-inflammatory, and that's probably we're gonna keep that for another discussion. There is botanical compounds, right? So things in plants that could actually have some anti-inflammatory effects. There is people working on this from a couple of different companies. Interesting area. But I guess Daniel and I, for the purpose of this presentation or this discussion, we're working on two main areas: vitamin D3, which is Daniel's work, and omega-3 fatty acids, which is work that we have in common and in some cases collaboration. So I'll probably like to start with vitamin D3 and ask you the question, why did you get interested? Just briefly, I guess. Interested in vitamin D3 in particular. We already figured this out, right? We should know what vitamin D3 requirements are. Why are we doing this? And how could it be helpful?
Well, the initial interest came from actually human research and experiments using mice as a model for understanding what happens during inflammatory bowel disease. So intestinal inflammation, there's a lot of that. And it was in the interest of applying those concepts to a leaky gut under heat stress. So it looked at from this mice data that when you have higher concentrations of vitamin D in the diet and you combine it even with calcium, you can potentiate in a way the response, reducing inflammation and improving health outcomes of these animals. So that concept should apply to the cow. There was not a lot of research at that time on vitamin D, specifically on the immune response. So we got interested in that for the heat stress. And I guess the larger point, if we're going to talk about nutrients, is that we could have an optimal concentration of nutrients in the diet. We say that there's adequate levels now for the animal, depending on the stage of life, the production level of the animal, etc. So we find those optimals, and we have the things like the NASEM or previously NRC that tell us at this level you're not going to encounter any problems. Animals are healthy. That is based on a few variables that you can measure at the time. Maybe it is production, depending on the nutrient. So if animals are producing well, like there's no issue. This is the concentration, and you should avoid deficiencies. You should avoid excess. Formulate diet for the typical animal, and that's all well and good. That the way it works is kind of like that. If we took the example of vitamin D for a second, we think about it in terms of how good it is for helping calcium metabolism. Like, for example, if we want to prevent hypocalcemia in early lactation, that's a really good way to do it. Bone formation, etc. The immune side is also part of it, but it's not often discussed, or as far as I remember, it was kind of new to me, the importance that it can have. So we started doing that research, keeping in mind that people already formulate diets to have higher concentrations of vitamin D relative to requirements or recommendations. And in the case of vitamin D, it's around 2 to 3x, 2 to 3 times more above the requirements. And people do it, field nutritionists, they know why they're doing it. There could be degradation during storage, and there's variation in the intake of animals. So you want to guarantee that everyone is around that optimal, even if they eat a little bit less, and you don't want to have that excess again. But it's not really optimized, as far as I know, for this nutrient or many other nutrients that we are interested in, for periods when the animal is stressed or diseased.
So say the requirement for stress, the requirement for disease states, not
And in
the
this case,
requirements for when everything is okay or acceptable.
right. So maybe heat stress, the requirements could come up, and this is something that we discussed previously. Animals eat less. So let's say a 30% reduction under moderate heat stress. Animals are going to ingest less vitamin D and less of other nutrients that may influence the immune response. So for that particularly important challenge, do they have enough? Maybe we have to boost it, the concentrations in the diet. And
But you
that's
don't,
the idea.
sorry, you don't boost it by 30%, that is accounting for the intake loss. You do it more.
Even more. Yeah,
So
we have an experiment where we have gone to, well, 4x. In this one, for example, in the heat stress experiment, we went up to 3.6 above the requirements. And just briefly, we see that animals have lower rectal temperatures, lower respiratory rates. So there's an improvement in the conditions of the animals under heat stress. So that would be the yellow group here. And what I'm showing in the graph is association of that temperature with the with TNF-alpha. I mean, over time, TNF-alpha, which is an pro-inflammatory cytokine on this pathway that we can see here that responds to LPS, is also decreased, the same as it happened for the rectal temperatures and the respiratory rates. On day 7 and day 14, there is a big reduction in the concentrations of this cytokine. So that could explain the results that we have for the hyperthermia alleviation and the respiratory rates. These animals are actually doing better. In this case, we didn't really see a response on productivity, but we have data that show, I think, that the animal is doing better. So we're helping cows with a relatively small modification of the diet, maybe targeting a particular time of period during the year to give this type of supplement. It would be the way to go. And then we followed up on this on a different model. So let's not focus only on heat stress. There's other scenarios where the animal can have endotoxemia, so that LPS going into circulation. So here we have a dose response experiment, and the preliminary data that we have so far tells us that cows at the highest levels of vitamin D, so 4x the recommendation, have lower rectal temperatures during an LPS challenge. In this case, intramammary LPS challenge, which is kind of creating a mock mastitis. You could do it in the uterus, different places where you could put the challenge, and it will tell you a slightly different story. But in the end, is how the immune response is being modulated by your diet, in this case by vitamin D. We also see that these animals have lower reductions in intake and lower reductions in productivity. We're yet to see what happens with gene expression of these immune cells, somatic cell counts, and all of this. This is very fresh data that we have.
But overall, what the story seems to be telling us is cows do better when you boost their vitamin D3 in the diet, which some people do intuitively, I guess. But there is confirmation here that in particular periods of stress, in this case, heat stress or immune challenge, you could do something about it.
Correct. I guess that's the take home for this.
Okay, I will just point out now to the similarities that we have in terms of the other part of our research before we end up the omega-3s, because there is overlap when it comes to this, which is again the activation of the immune system. So let's go to that as we finalize.
Maybe focus here on this pathway, the NF-kappa beta pathway. This is something that's going to come up also for the omega-3s. That you can see here is being blocked in a sense. So cytokine production and some lipid intermediates that result from this cyclooxygenase pathway are going to be different. So that's the interest in the omega-3s. So something
So
very
when we talk about omega-3s, just briefly, we're talking about things that the cow already has in their diet. For example, pasture. Fresh pasture is a good source of linolenic, alpha-linolenic acid, but also some things that are actually the longer chain omega-3s or VLCs that you should hear from fish oil, from algae, right? So those things in many animals, including the cow, seem to be good at reducing inflammation on two different sides, the initiation of the response and the resolution of the response. So that's kind of like why we are interested in this. And we both have some data on this. So probably just finalize briefly discussing what we have found.
I think that's a good point. The two are doing complementary actions. Sometimes they do the same, for example, for this pathway. But I guess that's why we have some interest in doing follow-up experiments where we have the additive effects of both. That's one thing that we want to really get into, to get some funding for doing that. So maybe just briefly show that during heat stress, we tried giving cows omega-6 or omega-3 fatty acids. This was our meson infusions. So we got recovery of production by 2 kilograms, so around 4.4 pounds, something like that, by giving just the omega-3s versus the omega-6.
Which is corn oil. You basically did fish oil versus corn oil. When you give fish oil, you get 4 more pounds, right?
Tuna oil versus corn oil. So omega-6 versus omega-3, and particularly long chains, EPA and DHA. So we get that alleviation of the rectal temperature increase. To some point, it was around 1 Celsius degree, which is a lot. It's like 2 Fahrenheit. I think we said that earlier.
Yeah, it's like going from a fear of 104 to 102.
Correct. So that's an important decrease. And on top of that, we see that it's associated with inflammation. So this is another indicator of inflammation that we mentioned earlier. So lipopolysaccharide binding protein is decreased in these animals compared to the animals receiving the omega-6. Omega-6 in red, omega-3 in yellow. So that came with the increased productivity and changes in metabolism that we don't need to go into right now. But insulin sensitivity was also improved.
Yeah, we could discuss that if there's any questions, but it seems positive. I guess that's the main take home there. We just to finalize with my part of this is that we did similar studies in which it's not heat stress, but we basically do the inflammatory challenge with the lipopolysaccharide again. And we can skip over this, Daniel, just to finalize. So we compared algae versus fish oil versus a very prominent popular fatty acid, which is oleic acid. Shown here is OA. And so these are potentially all beneficial for health in many animal species. But what we saw here is a couple of important things. Algae, which is a good source of DHA, is a very, very good source of DHA, as I said. Increased milk yield, reduced rectal temperatures when the cows were experiencing this heat of the inflammatory challenge, and interestingly reduced the somatic cells. So in terms of milk quality, also seems to be doing something because the neutrophils, which are cells that are infiltrating into the milk, actually are reduced in numbers. So it's telling us a story about the immune system freaking out a bit less, and that resulting actually in positive effects for the cow and for the quality of the milk that the cow is making. And all of that, again, accompanied by reductions in inflammation, which is borne out by other research in which we see that omega-3 supplementation, particularly very long-chain omega-3s, you can click on that, Daniel, it's an animation, I think, reduces a number of things related to inflammation. So it seems to be going in that direction in which the diet, in this case omega-3s, can do something to improve that. And that's true for algae or fish oil, but also flax seeds, depending on the study you look at. So very promising, I think, stuff happening in this area now.
Agreed. I guess, Alison, we're done with this section.
With everything.
With
Yes.
everything, I guess. Well, yeah, we don't have much time, and we have very good questions. So, again, thank you so much for all this interesting presentation and all this important information for everybody. So I'm gonna start with the questions that we have here. So the first one is, how can systematic inflammation influence feed intake? And no, go back, please. Thank you. Okay. How can systematic inflammation influence feed intake and exacerbate negative energy balance in dairy animals?
All right, so I guess the short answer to that, I should say, is inflammatory signaling in the brain reduces appetite. So that's a very predictable response. If there is inflammation, there is going to be a reduction in appetite. If you get that appetite reduction in a moment in which the animal is already struggling to get enough energy, which is again is the onset of lactation, you're going to have a more dramatic situation for the animal. So you're going to make it spiral down more likely. So yeah, probably that answers the question. It's not a good combination. If you have an inflammatory challenge, that's going to reduce appetite for sure. And we know that cows that show early reductions in appetite, and we had that slide somewhere, but early reductions in appetite tend to have a higher propensity for developing things like ketosis later on, right? So that's again going back to the marker that you can measure 3 weeks before parturition, already inflammation showing there, say haptoglobin, and then the cow getting sick later, right? But that started somewhere far away where you could not see it. So that's very important. It's going to make things worse in terms of appetite.
Okay, we have another one. So the million-dollar question: what comes first, inflammation, fat tissue mobilization, or drop in DMI? And the other one is, and perhaps a better question, how soon should we start assessing this physiological challenge? 21 days before calving, dry off? I would argue that we should look at even before the dry off.
Oof.
Okay.
Yeah. Maybe let me start with a few things that come to mind when we think about this. We have discussed this plenty with Eduardo because I
Off
used to
camera.
think there's one major thing. One major thing. There's the drop in the mammary intake around calving and the mobilization of adipose tissue that is even greater in cows that are overconditioned. And these are things that have been known for a long time. There's a series of markers before parturition that you could look at different times, 7 days, 14, 21 days. Thinking of maybe the end of lactation as well, the previous lactation. That's a point that I would say is also important to know, to characterize. But there's one thing that I really think makes a strong case for inflammation coming first, the chicken or the egg question, is that there's also some data, for example, from Michigan State, from René Sordillo's group. As far as I remember, it was some observational data, but it showed that animals that had lower concentrations of vitamin D in plasma before parturition, and I have to remember yet if it is 3 or 4 weeks before calving, those animals were more likely to be sick in the postpartum period, in early lactation. And that was an independent marker, independent of other things. So a very good marker of the propensity to be sick later, related again to the immune system, as probably we discussed enough today. So I think the inflammation part seems to be key. And the Andres Contreras from MSU data that Eduardo showed as well, it says, in my opinion, that most cows that are really sick have an inflammatory issue. It's not all of them, but the inflammation seems to be like the root of this. So now, Eduardo, I don't know if you have.
I will just add a brief point. They all may be happening normally. All cows are normally going to develop ketosis to some extent, that like all mammals, any animal that makes milk develops negative energy balance and ketosis. So I will just point out that that's a normal response and it will happen. And maybe the drop in intake around calving that happens, or parturition in other species. That's all to be expected. What we could do is to try to measure early in advance so that we know which animals on top of these normal responses, physiological responses, is going to get a more dramatic negative response on a negative trajectory early. So the question was 2 weeks, 3 weeks, 6 weeks, a dry off. Right now, we don't really have a good answer, but we should be measuring trajectories for sure. So I will be an advocate to measure as early as possible, because every time that we expand that frontier to, early as possible, we find that these markers keep on changing early. So I think that we need to answer that better, and I will put money on saying, well, $10 maybe, not 1 million. That the further we go back and before parturition, the better it will be, at least to know where we are. Yeah.
Okay, we have 2 more. So, okay, so you talk about fibrosis and reduction in milk production. In this part of the mastitis process, when we have first infection, then inflammation, and then fibrosis. Can you talk about the fibrosis, please? Also, are there nutritional recommendations for cow with mastitis or to prevent mastitis?
Well, I will have to admit not knowing details to that level of depth that would require to explain the fibrosis, other than an exacerbated chronic prolonged immune response is going to result in tissue damage. So we're talking about cells dying and that place being occupied by others, which is a collagen-like tissue that is part of what we call the fibrosis there. So that's as much as I can explain without really being able to go much deeper. So that's something you could avoid to some extent, not by avoiding the infection. If the environment is terrible, you cannot really avoid the animals getting sick. You need to do good management. But if you're there, what we're trying to make the case for is preventing through arming the animal with better defenses is going to be key. So that goes to the nutrition part. So hopefully that's good enough.
To quickly add to this, I know it's not part of the question, but it makes me think. We're trying to also improve the quality of life of animals. So well-being is one concern, welfare. So when they have something like mastitis and it develops all the way to fibrosis, there's many stages, of course. One of the things that we know is that there's edema, there is inflammation, there is pain. And with some of these therapies, we can reduce pain. For example, if we give the omega-3s, we're going to end up with a series of prostaglandins, for example, prostaglandin E3, that is anti-inflammatory and it reduces the pain feeling of the animal compared to prostaglandin E2. So there's things beyond that process that makes cows produce less, and it's also the welfare part. I think we can do a lot with that in this type of scenario where we help cows with nutrition.
Okay. Well, thank you so much. And just for outside research, what are the practical applications for managing inflammation? What do you guys think?
So strategies that we could use tomorrow at the farm. I guess that's
Let me start with one point before you get there. The first thing that we didn't really mention a lot, we would remove this slide because of time, is management. Doing the basics well, with many farms do already. It could be as simple as not feeding an animal the same feed for 2 days or 3 days in a row during the summer months, like simple stuff like that, which sometimes happens. It could be social interactions, proper group management. There's a number of things that we know already, and we don't need this research to tell us that you could do better for the animals. And that all seems to result in reduced inflammation. So inflammation is going to be normal, but you don't make it go as wild if you actually handle the basics well. Now we're talking about something beyond the basics, which is saying, okay, give more omega-3s, and we can discuss, because this is a costly thing, or give more vitamin D3, probably a more cost-effective thing to do. But there is a number of things outside of the normal, but the normal has to be, the regular management has to be on point. That probably will be the first thing that we should fix, the base of that pyramid, before getting into the more expensive stuff. That's my take home, I guess.
Agreed.
Okay, well, let's go to the next slide. Thank you so much. Join us next month for the Badger Dairy Insight on August 19 at 11 a.m. Our topic will be adopting and evaluating mechanical ventilation in dairy barns, a calculator guide. Find more information at the Wisconsin, at the UW-Madison, Wisconsin extension website. You can go to the next one. Scan the QR codes. We have here 3 different QR codes. You can go the first one for the dairy website. You can find a ton of different information, research. The next one is the Badger Dairy Insight that you can sign in for the next following sessions. And the third one is for the dairy e-newsletter. So you have it monthly, all this information about what is happening in the dairy industry and the dairy academia. And the last one, well, for additional information and unbiased, please, the university have extension dairy program online. So you can go to the dairy, go to the next one. Go to the dairy.extension.wis.edu or find us on Facebook at Extension Agriculture. Thank you so much, everybody, to be here. Thank you, Daniel. Thank you, Eduardo. Really appreciate your time. Thank you, everybody, for stay here for 3 more minutes. I hope you guys enjoyed this big conversation. I know we just missed some things, but that was very, very important. Thank you so much, everybody.
Thank you.
Thank you.
Yeah, and you guys are the best brothers, twins ever.
Thank you.
And okay, thank you so much, and have a great day, everybody, and have a beautiful evening. Thank you. Bye-bye. Okay, I think that's it. Thank you so much.
Good. All's done.
Thanks. We tend to talk a lot. We try to measure ourselves, but anyway.
It's okay. It's okay. I think yeah, I think it was good.
All righty.
Daniel needs to talk a little bit more, I guess. He was a little quiet.
No, I was trying to be the host to Eduardo.
Oh, okay. Okay. I can see that now. Okay, well, thank you so much, and we can talk later.
Yeah.
Yes, awesome. Thank you for having us again. It was a cool exercise, so appreciate it.
Yeah, no, thank you so much. Have a great day.
You too.
Yeah.
Bye-bye.
Bye.